Welcome, everyone. Happy Friday. I'm Tanya Stevenson, the CEO at MDF, and I'm delighted that you are all joining us for the launch of our 2024 Meet the DM Drug Developer Series. This is the fourth year that we are hosting this series, and I'm happy that we are able to invite Dyne Therapeutics to join us today and talk about their progress towards a treatment for myotonic dystrophy. Before we jump in, I would like to first thank MDF's Director of Technology and Communication, Clede Cumming, for making this series come to life over the last 4 years and ensuring that we are always posting this to the Digital Academy and making it accessible for our community. I appreciate you, Clede, and I'm very grateful. Now, it is my great pleasure to introduce to you MDF's new Chief Scientific Officer, Andy Rohrwasser. Andy brings with him a broad and diverse background in academic science, venture-based startups, and public health. Andy's domain expertise and passion are rare diseases, genetics and genomics, and newborn screening. He serves as an adjunct associate professor in pediatric neurology at the University of Utah, where he also received both his PhD in human genetics and his master's in business administration. Andy, welcome. We're so grateful that you are here with MDF. And please take it away.
Thank you very much for the very kind introduction, and welcome everyone. So as Tanya said, welcome to our first webinar of 2024 in the Meet the DM Drug Developers series. Today, we welcome Dyne Therapeutics and their presentation entitled Achieving the Promise of Force to Deliver for Patients. Next slide, please. So a little bit about the Myotonic Dystrophy Foundation. We, of course, envision a world with treatments and a cure for myotonic dystrophy. The mission of the Myotonic Dystrophy Foundation is community, care, and a cure. So we support and connect the myotonic dystrophy community. We provide resources and advocate for care, and we accelerate research towards treatments and a cure. We also provide resources and support. Many of these resources you'll find on our website, myotonic.org. So toolkits and publication. Here, MDF has convened leading experts in the area of myotonic dystrophy to create resources that guide care providers and families in the management of the disease, and they include anesthesia guidelines, the MDF toolkit, and clinical care recommendations. We also support the support groups. So these groups are led by trained community volunteers. And these programs create safe spaces to network, to learn, and to share. On our website, you also find the Digital Academy with hours of educational and inspirational videos by DM experts on demand. And that's also the place where in a week or so, you will find a recording of today's webinar. Last but not least, there's a calendar that contains all upcoming support group events, conference, webinars, and more. And again, you find all of that at myotonic.org. The big news of 2024 is that we are holding regional conferences. So the point of these conferences is that we want to come to you, to your regional communities. The first conference is on March 23rd in Gainesville, Florida. The next one on April 6th in Boston. The third one on April 20th in Iowa City. The fourth one in Houston on May 4th. The fifth one in Los Angeles on May 20th. And the last one, June 1st in Seattle. Registration is free. You find it again on our website, myotonic.org 2024 MDF Regional Conference. And in 2024, we don't hold our annual conference. We will resume that in the spring of 2025. We will resume the Meet the DM Drug Developers webinar series in the fall of 2024, so after the regional conferences. You find the schedule right on our website again, myotonic.org, Meet DM Drug Developers. So please sign up to get latest updates in our monthly e-newsletter, and you again sign up through that on our website, myotonic.org, sign emails. And this is hot off the press. We already have the next company lined up, Dyne Therapeutics. They will present in July also on a Friday. So let's talk about drug development in myotonic dystrophy. When I think about drug development, I see many parallels to soccer, which is one of my favorite sports. And one of my favorite teams is Bayern Munich. And as you might know, their top scorer is Harry Kane. He was also the captain of the British national team. And what you see here, you see a graphic representation, the dark red spots. These are positions from which he scores goals. And then you have these kind of lila purplish open circles. They represent unsuccessful attempts. So why do I tell you about soccer? Well, similar to DM, lesson number one, in order to make goals, we have to shoot at goal. And we have to shoot the ball onto the goal from various different positions. So the more we shoot, the higher the probability of success. And that is also the case in myotonic dystrophy. Next slide. So we can broadly distinguish between 4 areas in the drug discovery field. They represent the basic dogma of biology. So where we have DNA with the genes, then we have a process called transcription to form RNA. And in the disease state, we have these repeats. So the result is RNA misfolding and the formation of these toxic hairpin loop. These hairpin loops, they act as traps and they capture important cellular proteins. And when they are not available to do their stuff, we have malfunction. So the first category of drug discovery activities focus on the inhibition of transcription and gene editing. The second target inactivation of toxic RNA. The third one aims to compensate for these trap proteins, and the last one broadly tries to compensate for and to correct cellular dysfunction. A brief word on DM and DM1 and DM2 research. While area 1 and 2 are disease specific, area 3 and 4, they have the potential, right, to have commonality. So lessons learned, for example, in a DM1 trial, they might or they are actually of immediate relevance for the DM2 community. Next slide. So today we hear about Dyne Therapeutics and their activity, next, focus on the inactivation of toxic RNA. So it's our great pleasure to introduce Ash Dugar. Dr. Dugar is the Chief Medical Affairs Officer. He brings to Dyne over 20 years of broad experience across medical affairs, clinical and commercial development, and real-world evidence generation. Before joining Dyne, Ash served as a VP, the Vice President and Global Head of Medical Affairs at Sarepta Therapeutics, and in various leadership roles at Intracellular Therapies, Roche, and Pfizer. Ash began his career at the NIH, the National Institute of Health. He completed a pharmacoeconomics and outcomes research fellowship at the University of Michigan School of Public Health. He earned an MBA from Penn State and a PhD in pharmacology from the Pennsylvania State University College of Medicine. So before we hand it over, a few housekeeping issues. So we can ask questions live. You, the audience, can ask questions live. So click the Q&A button to open a window, type your response to the question in the text field, and you can send your questions anonymously. And then click send, and we capture the questions. Next. So without further delay, welcome Dyne, welcome Ash. Thank you for joining us, and we are looking forward to hearing about your results.
Great. Thank you so much, Andy, for that very nice introduction. and definitely appreciate the overview of the disease and various modalities to tackle the disease. Like you, we hope that all of the investigational medicines that are being developed, we hope all of them make it to market. And we certainly need a lot of shots on goal, to your point, to make sure that there are some that do, that can really change the trajectory of the disease. So as Andy said, my name is Ash Dugar. I'm the Chief Medical Affairs Officer at Dyne Therapeutics. First, I'd like to thank Tanya and the entire MDF organization for the opportunity to present an exciting update on our DM1 program today. Before I begin, I'd like to introduce you to our new head of patient advocacy, Catherine Beaverson. As many of you know, Catherine has been part of the neuromuscular advocacy community for many years and brings tremendous experience and passion to this role. So I'm so pleased that she's joining, and she'll come back when we do the Q&A session. Importantly, please know that Molly White isn't going too far. She was the head of patient advocacy. She's taking on a new role here at Dyne. So I'm super excited for this addition to our team with Catherine's arrival, while being able to work very closely with Molly in her new role. And I would be remiss if I didn't thank Molly for building Dyne's key pillar, which is our patients first core value, and connecting Dyne to this incredible community that we serve. So thank you, Catherine. Thank you, Molly. Next slide, please. Before we begin, I'd like to remind everybody that I will be making forward-looking statements today, and these are subject to safe harbor protections. You can go to our website if you would like to read more about this. Next slide, please. So at Dyne, it all starts with our mission to deliver life-transforming therapies for people living with serious muscle diseases. Our mission is woven deeply into our core values and inspires our employees, dynamos, as we call ourselves, each and every day. Next slide, please. A little refresher on our scientific platform. You've heard me talk about this in the past. The intentional design of the FORCE platform is really translating in the clinic, both from a safety and what we call a pharmacodynamic perspective, as we're seeing across both of our clinical trials. So we had a hypothesis. We tested that hypothesis in preclinical models, and the data that I'll go into shows that that translation from preclinical to clinical seems to be holding up in the ACHIEVE study for myotonic dystrophy type 1, and also in what we call our DELIVER study in Duchenne muscular dystrophy. So by way of reminder, the FORCE platform consists of 3 components. Here you can see on the left-hand side of the slide, the antibody fragment, or the Fab antibody. It's designed to bind tightly to the transferrin receptor 1, which is highly expressed on the surface of muscle cells. This includes skeletal muscle, diaphragm, cardiac, and smooth muscle. At the bottom is our linker. This is a clinically validated linker, which we chose because of its demonstrated safety and efficacy in third-party approved products. And on the right-hand side is our antisense oligonucleotide in the case of DM1, or we can attach other kinds of payloads. So we select a payload that is used to target the genetic basis of disease. And this is where we leverage the modularity of the FORCE platform. So what we mean by that is for nuclear targeting diseases such as DM1, where we know we have to degrade toxic DMPK in the nucleus, we choose ASO or antisense oligonucleotide payloads. For diseases that are more cytoplasmic in nature, where the target resides in the cytoplasm, we can choose something like an siRNA, for example. And we also have the potential to attach small molecules or even enzymes with the FORCE platform. So it's highly versatile, allowing us to adapt depending on the genetic basis of the disease. Next slide, please. So I know that you know this disease far better than I do, and we've actually been learning tremendously from the community. Just very briefly, as you know, myotonic dystrophy type 1 or DM1 is a devastating disease with no approved treatments. It's an autosomal dominant triplet repeat expansion disease of the DMPK gene. In DM1, the higher than normal number of toxic CTG repeats, it forms large hairpin loops that remain trapped in the nucleus, these kind of trapped proteins, if you will. And it forms nuclear foci that then contain or trap the splicing proteins that lead to abnormal splicing in many genes. This abnormal splicing, or what we term spliciopathy, results in a wide range of symptoms, often characterized by onset of muscle myotonia, which is the inability of muscles to appropriately relax after contraction. This is followed by weakness and atrophy of skeletal muscles, and symptoms include cardiac conduction abnormalities, GI and metabolic disturbances, respiratory distress, as well as CNS manifestations. Next slide, please. The FORCE platform harnesses the natural mechanism of TFR1 receptor mediated delivery to transport therapeutics across the cell membrane. Now, this is differentiating because we're not using membrane destabilizing agents to deliver the antisense oligonucleotide into the cell, or the way in which it leaves what you see here called the endosome. So we have the conjugate that goes into the cell, forms in this, or it is contained within this endosome, and then the antisense oligo is cleaved within the endosome and travels to the nucleus so it can do its job. Next slide, please. So Dyne is well on its way towards achieving the promise of FORCE in delivering for patients. We've delivered exciting proof of concept clinical data for our DM1 program, and I'm really pleased to share those initial readouts today. Next slide, please. Thank you. So our trial is informed by multiple stakeholders. We have gone to various key opinion leaders across the globe and regulators to get input into our overall design for the ACHIEVE study, and this is in patients from 18 to 49. We've also gotten wonderful input that help define the outcomes measures that we have in our study, as well as the biomarkers. So these include splicing, myotonia, measures of muscle strength and function, and of course, key safety considerations. Also, we have learned a tremendous amount from the key opinion leaders on the natural history. This is critical so that we can contextualize the clinical trial data that we are generating and we will continue to generate. We've also gone to many of the advocacy leaders, such as MDF, around the world to help inform our clinical trial as well. This includes considerations for trial selection, input into the clinical trial protocol and visit schedule, and what we can do to minimize patient and family burden during trial conduct. And we've also gotten tremendous input on various ways to ensure that we provide adequate support throughout the clinical trial process and education that patients and families will want to have. Next slide, please. So before I go into detail on the data, I just want to review the ACHIEVE study design very briefly. ACHIEVE is a global trial, and we have designed it to be potentially registrational. The primary endpoint is safety and tolerability, and a key secondary endpoint is splicing, a pharmacodynamic endpoint. And we're evaluating a number of other functional assessments. It is a 3-part study. There's a 6-month placebo-controlled multiple ascending dose portion, or MAD, a 6-month open label extension, and a 96-week long-term extension. Importantly, we designed ACHIEVE to be adaptive in nature so that we can optimize the dose and dose regimen within the trial. And this is all part of our efforts to move as quickly as possible to advance Dyne 101 in the clinic and eventually with regulators. Our goal is to first show an improvement in splicing, and then over time to be able to correlate that to changes in function. That is what we believe, based on our conversations with global regulators, opens the door to potential expedited approval pathways. Next slide, please. So on this slide, you can see some of the main inclusion and exclusion criteria. For the full list, you can please go to our ct.gov page or UDRA-CT page, which also contains a list of all of our sites for this global study. And those identifying numbers are located at the bottom of the slide. So now I'll go into the initial results that we presented previously or earlier this year. Thank you. Very importantly, we're very pleased with the safety profile for Dyne 101 that we've observed in ACHIEVE so far as of the data cutoff date. Now, we believe this is very differentiating from others in the field and speaks to the importance of each component of the FORCE platform, as I mentioned earlier, the antibody fragment, the clinically validated linker, and the choice of the antisense oligonucleotide. This safety data is based on 45 participants dosed through the 5.4 mg per kg cohort. During the MAD portion of the trial, most treatment-emergent adverse events were mild to moderate in severity, and they typically occurred around the time of infusion. There were 2 reported serious adverse events. Both were deemed unrelated to study drug. One was a case of pneumonia, and the other a transient worsening of atrioventricular block in a participant with ongoing medical history of first-degree AV block. Unfortunately, the natural history of DM1, in the natural history of DM1 patients, there's a known background of arrhythmias. Liver enzyme elevations were seen in about 18% of the patients. But importantly, no impact on liver function as measured by bilirubin occurred. We also know that liver issues are a part of the natural history in DM1 patients. Importantly, there were no reports of anemia, thrombocytopenia, or kidney injury. So approximately 300 doses have been administered and ACHIEVE across the MAD portion and the open label extension, and the profile has supported dose escalation up to 6.8 milligrams per kilogram. So we're really pleased with the safety profile, which allows us to continue to escalate the dose. Next slide, please. So I'm going to talk very briefly about the PK, PD, and functional data, but take a deeper dive into splicing specifically. So the placebo patients are in the gray bar, 1.8 milligrams per kilogram cohort in blue, and 3.4 milligram per kilogram cohort in green. The key takeaway from this slide is that we see dose-dependent delivery of our investigational medicine to muscle, DMPK knockdown, and splicing correction in 3 months. So this is a really important part of this investigation, that we can see dose-dependent delivery to muscle, that DMPK knockdown, and splicing correction at 3 months. If you go to slide 13, please. I'll go into more detail on splicing specifically here. As I mentioned, we see clear dose-dependent improvement in splicing in 2 ways. First, we see a greater improvement in splicing with the higher dose. 19% improvement in splicing among the 3.4 mg per kg cohort versus 13% for the 1.8 mg per kg cohort at 3 months. Second, we see all participants respond with the higher dose. We have 5 of 5 responders among the 3.4 milligram per kilogram cohort versus 3 out of 5 for the 1.8 milligram per kilogram cohort at 3 months. And we see consistency both in the number of responders as well as the quality response across what we utilize, which is a 22 gene panel. It's the same splicing panel that has been used in other studies. And also what's exciting is that in our 3.4 mg per kg cohort, we're on the cusp of the range of improvement in splicing that we hope to see over time and ACHIEVE. We've said for some time that based on our own preclinical work, input from others in the field, and the natural history, we believe 20 to 25% splicing correction is needed to drive functional benefit for patients. And because Dyne 101 targets the nucleus where this missplicing manifests, we believe effectively knocking down nuclear DMPK will drive disease modification, and we believe we're beginning to see that. Also, a little more color on the 1.8 mg per kg cohort. 1.8 milligrams per kilogram was selected as our starting dose because we believed it would be a pharmacologically active dose based upon our preclinical data and modeling. And this is what we're seeing with these data. First, we see an overall improvement in splicing at 3 months, and we see that same 3 of 5 participants demonstrate improvement in splicing at both 3 and 6 months. Now, the increase in our CAS or splicing score between 3 months and 6 months among participants in the 1.8 mg per kg cohort is really driven by worsening observed among the 2 non-responders. So at 6 months, you see the 7% increase in splicing. And again, that is driven by those same 2 non-responders that we saw in the 3-month timeframe, and then again in the 6-month timeframe. Because this was the lowest dose, we're not surprised to see heterogeneity in response, similar to what we see in the natural history. Again, we predicted the 1.8 milligrams per kilogram to be our pharmacologically active dose, and we are clearly seeing data that support that. Furthermore, we're seeing benefit on function even at this low 1.8 mg per kg dose, and this benefit extends out to 6 months. And all of this is inconsistent with placebo. Next slide, please. So here we see meaningful improvement in myotonia. And as I mentioned before, it's the inability of muscles to appropriately relax after contraction. We see that with treatment with Dyne 101, we demonstrated an improvement of nearly 4 seconds at our lowest 1.8 mg per kg dose at 6 months. The belief all along has been that myotonia may have the best opportunity to improve quickly with an effective therapy. So for this early read, we thought it was reasonable to assess myotonia via video hand opening time, or VHOT, at 6 months. This involves asking the participant to make a fist and then opening it. A central reader evaluated the time it takes to open the hand. On the Y, or vertical axis, it shows the average of all the myotonia trials for an individual participant in the trial in seconds, and the horizontal bar, or the X-axis, is the duration during the trial. As a reminder, we are only showing 1.8 mg per kg cohort because we have the full 6-month data. You see a clear worsening in the gray placebo line, and in the Dyne 101 group, improvement early that grows to a 3.8 second improvement from baseline. These are very striking data, and we're excited to see how this and other functional measures evolve over time and at higher doses in ACHIEVE. Next slide, please. Finally, we're pleased to share an early look at a patient-reported outcome, the Myotonic Dystrophy Health Index, or the MD-HI. The MD-HI is comprised of multiple subscales, including fatigue, strength, GI health, cognition, and the ability to do daily activities. Patients in the 1.8 mg per kg group experienced an overall improvement in MD-HI, and these patient-reported outcomes data are very encouraging. And we look forward to further evaluating PROs as part of our commitment to comprehensively address DM1. Next slide, please. So in summary, these initial data show robust effect of Dyne 101 in DM1 at our initial cohorts dosed once a month. Our goal in this initial read was to begin to see an effect that is separating from placebo, and we are thrilled with these compelling data. We see dose-dependent increases in muscle delivery, DMPK knockdown, and splicing at 3 months, as I mentioned before. Consistent splicing correction across patients in the 3.4 mg per kg dose. Meaningful functional improvement in myotonia as measured by VHOT at the lowest 1.8 mg per kg dose. And a favorable safety profile. The 5.4 milligram per kilogram cohort dosed every other month is already fully enrolled. And we have 48 patients enrolled across the trial with approximately 300 doses administered. We believe we achieved proof of concept with these early data, and our focus is now on optimizing the dose and dose regimen this year. We believe we're well on our way to driving the splicing correction that we feel is needed to drive comprehensive functional improvement. And we anticipate sharing our next update from Achieve in the second half of this year. Next slide, please. So I have to thank all the families, patients, investigators, and their teams for the sacrifices they've made to advance therapeutic options, including Dyne 101 over the years, over the decades. We know there is significant burden that all of you take on to progress the science and advance medicine. Thank you so much. And once again, thank you to MDF for having us here today. I'm happy to take questions.
Fabulous. Thank you, Ash, so much for this for your presentation, right? And the positive data you are presenting here. So we have questions.
And Andy, sorry, may I ask Catherine to join me here on the Q&A? Thank you. Great. Super.
Of course. So
Okay.
we have a couple questions here. Let me go through them. So if you were to move on to a phase 3, would you increase the age of inclusion?
Yeah.
And then similar,
yeah.
maybe we can combine those two. Can you also comment on the initial age group limitation to 49 years of age?
Sure. Happy to. So maybe I'll start with the second question first because it informs your first question. What we have learned from the families that have partnered with us and with the key opinion leaders is what I think everybody knows. There's a tremendous amount of heterogeneity in this with this condition and in this population. So in planning out our phase 1/2 study, as most companies would do, you take a very thoughtful approach in terms of what the early trials look like. You learn from those early trials, and then you expand as you move into the later stage trials, right? They need to become more generalizable as you get further down the path of drug development. So not just with respect to age in terms of inclusion exclusion, but in terms of much of the criteria that we specify for the Achieve study. The intent was to be able to have a population where we could absolutely feel comfortable about any sort of signal that we are detecting in terms of efficacy. So we wanted to have a population that was somewhat homogeneous, right? Trying to reduce the heterogeneity in this first study so that we can understand what Dyne 101 is doing, especially from a safety and tolerability point of view, and then also with respect to some of the functional measures that we've added. So we try to create a population that, for example, wasn't sort of at a ceiling, so there was no room to improve, and at the floor where we may not be able to kind of understand what's happening on the bottom end of function. So all of the inclusion exclusion criteria is really geared towards trying to create some sort of semblance of a population that we can feel good about putting Dyne 101 testing Dyne 101 so that we can understand what's happening with respect to safety, tolerability, splicing, and function. So trying to create some sort of homogeneous population within a very heterogeneous population. And then as we move into phase 3, we're going to learn. We're going to learn about our dose regimen. We're going to learn about what endpoints move in a reasonable period of time. We're going to take these learnings and then apply them to our phase 3 study. So right now, I can't tell you what the age range or other criteria will be that's different in our phase 3 study than it is in our phase 1/2 study, because we still need to generate more data, take those learnings, and then apply them to those later stage studies. But to your point, in terms of drug development, we want to make it more generalizable.
Great. So there are lots of there are several questions here in live questions as in questions that were submitted before during the registration. Can you just, in very, very high level and very general terms, comment on how trial site selection works? So because
Sure.
we get questions. When will the trial come to the US? When will the trial come to Canada? When will the trial come to Italy, and so forth? Maybe you can shed some light into that.
Yeah, sure. So first of all, this is it. Just Achieve itself is a global trial. Right now, all of our sites are in Europe and New Zealand. In terms of how site selection is done, probably need half an hour, a whiteboard to really go through it. But to be perfectly frank, different companies approach site selection differently. If I think about the myotonic community specifically, a first approach to site selection is engaging sites that have experience running clinical trials in myotonic dystrophy. And one of the core places to go to understand which sites are those is in the DMCRN network, so the Myotonic Dystrophy Clinical Research Network. So there are a number of sites across the globe that are in that network. These investigators have a tremendous amount of experience running neuromuscular studies, running myotonic studies. They are all, I believe, involved in the natural history NDM1 study. And so they have the capabilities ready to go in terms of executing interventional trials. So a good place to start for companies looking to do trials in myotonic dystrophy is starting with the DMCRN. And then from there, as trials get bigger, as companies look to expand their site selection, they will look first to sites that have experience running neuromuscular trials and hopefully in the space of myotonic dystrophy. So it's sort of this approach of like, let's make sure that we have a core group that has experience. And as you need to expand your trial, run larger phase 3 studies, et cetera, you'll increase that scope. And then there are typical engagement processes, training processes, standardizations to make sure that all of the sites have the capabilities and are running the trials in the same manner so that you reduce variability from site to site, or you try to minimize variability from site to site. But at the core for the DM community, the DMCRN is a great place to start. Catherine, I don't know if you have anything to add.
No, I was just going to kind of expand on this because I think, Andy, if you don't mind, there are a couple of questions that are kind of lightly touching upon where Ash is going, which is kind of when we think about our future planning, future studies, right? You've kind of given us a good explanation of how we think about where to go. But there are some questions that refer to timelines, right? When we think about when is that next study? When does, the kind of go to market time point come in? And then, what is future thinking around other functional endpoints? Those are some of the questions.
Yeah. And sorry, Andy, Catherine triggered me for one other comment on site selection. There is a very specific process for companies to go through to open up clinical trials in countries. And then, of course, opening up specific sites in those countries. And, in some ways, that also drives decision-making for what sites are selected.
Perfect. So maybe let's wrap this trial inclusion and things up with one last question here. From a patient perspective, who wants to participate in a trial, what advice would you give to her or him?
Yeah, I'll start an answer, and then I'm going to ask Catherine to also chime in here. So I think the place that we always start when a family or a patient is exploring entering a clinical trial is please speak to your physician. They will be, your first partner in thinking through options for clinical trials. And in myotonic dystrophy, the choices are increasing, which is wonderful, but there's also more to think about and more to discuss with your physician. So please start with your physician in terms of thinking about what clinical trials might be useful to you. And then from there, the clinical trial sites are wonderful. They're great sources of information. Going to the sites in terms of calling them or emailing them is a great way to get some information directly from the site. And so they also are a great resource. And then finally, the companies themselves often have information and resources for those interested in clinical trials. And you can go to their websites or contact some people directly to get more information as well. But it does start with a conversation with your physician.
Great. So let's go into more technical questions with regards to the potential administration of your drug. So one attendee would like to know, how do you think the substance would be administered ultimately,
Yes.
if and when it gets accepted?
Yes, it is intravenous administration in the clinical trial, and we would anticipate that if we are granted marketing authorization, it will also be administered IV. And as I mentioned before, our trial currently is dosing every 4 weeks, and we are also exploring dosing every 8 weeks.
Perfect, thank you. So next set of question, if you wouldn't mind expanding a little bit with regards to secondary outcomes. So you target the muscle, right? One of the endpoints is improvement of myotonia. So would you mind speculating with regards to the improvement of, for example, pulmonary function, digestive function, cognitive impairment, fatigue, daytime sleepiness, and those issues? And we should really be clear, right? You made it clear in your presentation. You target the muscle. So with that, would you mind speculating?
Yeah, absolutely. So I'll speculate a little bit, not too much. So let me start with the outcomes measures. So we are assessing a number of different measures of strength and function. The full list of those, or nearly the full list of those measures, you can find on the ct.gov page, for example. So we are looking at things like 10-meter walk run, 9-hole peg test, etc. But to your point, Andy, we're also looking at pulmonary function. So we do have measures of pulmonary function as well. We also have measures of cardiac function. So we are looking at areas that we believe are appropriate targets for Dyne 101. In terms of areas like smooth muscle, so like GI disturbances, for example, we are looking to explore that in a more robust way. So we do have patient-reported outcomes, and we can get a little bit of information on GI disturbances through something like the MD-High. But we want to actually sort of take that to a next level. And so we are thinking about what we can do to even more directly measure GI disturbances. But we are getting at it from a patient-reported outcomes point of view. Similarly, in terms of the CNS effects, some months ago, I presented to this community our data, our preclinical data, showing that our conjugate gets into the brain, and where it gets into the brain, not just the superficial layers like the cortex, but also the deep brain, caudate and putamen. So we're really excited at the potential to be able to have some CNS benefits. And once again, we are going to be able to use the MD-High to get at some of that. The MD-High has measures like fatigue, for example. And in our initial data that we showed earlier this year, we actually showed an improvement versus placebo in the fatigue subscale of the MD-High. And it has other subscales, cognition, and et cetera. So the MD-High allows us to be able to get a peek into how we might be alleviating other components of the pathology that we can't maybe measure through biopsies, for example.
Maybe last question with regards to the muscle pathology. Would you mind speculating? There's a question here as well with regards to the impact on proximal versus distal muscle groups. Where would you see effects first, and what could you share here?
Yeah, so that would be. So let me back up. So we took biopsies. We do biopsies in this particular study, needle biopsies, and it's at the level of the TA, so that the lower leg. So we do believe that we'll have the opportunity to see benefits across different muscle groups. And again, it's something that we are going to understand over time. So if I relate it to splicing, within our splicing correction, there are a number of factors. One of those factors, for example, is the CLCN channel, which is known to be related to myotonia. And KOLs have said you may be able to actually see a faster change on that particular factor, and therefore see changes in myotonia before you see changes elsewhere in terms of strength and function. And indeed, we sort of are starting to see this in our initial data. So I think we will learn in terms of what kinds of benefits we have and where over what time period as we generate more data at higher doses and longer term data. So, and again, some of these splicing factors change faster than others. And so that may be related to where we see changes, in certain muscle groups versus other muscle groups over time. So again, I think my message is we're learning. I don't know right now how this is all going to pan out, but we're pleasantly surprised. We are really eager to see the measures of strength and function, in the second half of this year and see what that looks like. So we'll learn a lot more, and I think I'll have a better answer to your question later in the year.
Very well. So now let's maybe there are several questions. Let's look into the future a little bit. First, we of course represent all of myotonic dystrophy, and considering that your delivery system works so well, are you considering in the future employing this approach also to DM2?
Yeah, it's a question we get all the time. And so, right now our focus has been DM1. And I'm not trying to, I would like to directly answer your question, but we're learning. We need to still learn about how this alleviates or how this may benefit DM1 patients. But we are well aware that the technology might be applicable to type 2 as well. I just can't say that this is where we're going because we actually are still learning about what this conjugate can do.
So looking even further into the future and knowing that you have a well-functioning muscle delivery system, do you eventually see the employment of combination therapy, for example, where where your drug would be utilized with others, whether they are small molecule drugs or or drugs that interfere with global correction of cellular dysfunction?
Right. No. So again, it's an important part of the discussions we have internally. So this is speaking hypothetically because there's no data that exists to hang our hat on. But yes, I mean, I hope we're at a place where polypharmacy, where multiple therapies are available, and if so, I would have to imagine they will be used in this space. And there are analogs, not perfect analogs by any means, but one can see in in the SMA space, you see a combination of you have small molecules, antisense, and gene therapy, and they are used in combination to some extent. In the Duchenne muscular dystrophy space, there is a strong anticipation that multiple modalities will be combined in the Duchenne population, small molecules, antisense, and gene therapy. So similarly, we believe that, and we hope that there's the ability to have multiple products approved, and that this antisense therapy can be used in combination with other therapies as well, symptomatic or otherwise.
Right.
We just need to have more molecules and then generate that data.
Along the more molecule question, there's a very interesting question in the in the live submission here, and that is again, considering that you have a very elegant and effective delivery systems. Have you also considered the delivery of siRNA molecules with this system, or it's probably not something you can talk about?
Well, I mean, So as I think I mentioned on one of the slides, the modularity of this particular platform does allow us, for example, to attach an siRNA. So we have the ability to attack targets depending on where they reside in the cell. As is well understood in myotonic dystrophy type 1, and you sort of alluded to this earlier in your talk, there is the toxic DMPK RNA that resides in the nucleus. So our antisense, as antisense oligos do, they target the nucleus. So they get into the nucleus and tackle the genetic basis of disease. So that is why we attached an antisense oligonucleotide and use that for our conjugate to get to that toxic nuclear DMPK RNA. For other diseases, we may want to attach an siRNA because we want to get at the toxicity that resides in the cytoplasm. So absolutely, we've thought about it. We do believe, obviously for DM1, we have the right payload, and our initial data is supporting that hypothesis.
Great, thank you. Maybe a couple more questions, and I think you have already answered that. So, does your product cross the blood-brain barrier?
Yeah. So what we have more work to do. What we have shown, what I've shown some months ago to this audience, is that our conjugate does cross the blood-brain barrier. So we showed in non-human primates that a tool conjugate crossed the blood-brain barrier and got into the cortex, caudate, putamen, and we were able to show deep brain penetration. And we contrasted this with delivering IV unconjugated antisense, and also versus antisense delivered intrathecally, so directly in. And what we showed is with IV delivery, there's essentially no delivery to the brain with a naked unconjugated antisense. With the intrathecal delivery, what we showed is that we were able to get that antisense into the superficial layers, like kind of just into the cortex. And then with their tool conjugate delivered IV, we showed that deep brain penetration. And that was one model. In the other model, we used our mouse model that expresses the human transferrin receptor and human toxic DMPK. And in that mouse model, we were able to actually show a reduction in those clumped proteins and that trapped foci in the nucleus in different parts of the brain. So, again, we have more work to do, but we're excited by, at least initially, what we're seeing the force conjugate can do in the CNS.
Great. So considering that we are pretty close to the hour, right? So maybe if you could end with description of timelines, estimates, because that's what gives patients hope, right? In the end, how will the trials progress in the most optimistic sense, and when will we see the next phase and so forth?
Right. So obviously I can't give specific quarters and years. But maybe a few things to put some color around your question, or try to answer your question. We're having conversations with multiple regulators around the world in terms of how to expedite the development and approval of Dyne 101. That's number one. I think number two, we have moved very quickly from idea to having this initial data in hand to give us better sense of optimizing the dose and dose regimen. So really the goal for this year is to make sure that we can hone in on the dose and dose regimen for Dyne 101. That's absolutely critical, because then that opens up our door to really go full force and full steam in terms of testing that optimal dose and dose regimen with the proper outcomes, so that we can put this data in front of regulators as soon as possible and get marketing authorization. So, again, at the end of this year, or, late when I come back, hopefully sometime at the back end of this year, I'll be able to give you some more input in terms, or more sort of visibility into the status of the program, because optimizing dose regimen is really our key goal for the program right now. But we're trying to move as fast as we can. The investigators have been amazing. I mean, they've been working so well with us, because everyone knows there's such a huge need for more clinical trials, and then again, access as soon as possible.
So on that note, thank you very much, and of course we will take you up on your offer, right?
Anytime.
To come back, right, and stay in touch. And for the audience, if there are any really pressing question, reach out to MDF afterwards, and we will try to facilitate. So with that, again, thank you very much, Dyne and Ash. Great presentation, very clear. Thank you for your patience answering all these questions. So if today's programs did speak to you all, folks in the audience, please consider showing your appreciation with a donation. We can only solve that together, right? Only if we come together as a community, we can really change the future of this disease. The link to donate is again on our website, myotonic.org/donate. And the last point I want to make is I really want to plead with you all, please contribute to myotonic dystrophy research and join the myotonic dystrophy family registry and update your profile. This is really super important, right? To facilitate future clinical trials, natural history studies, because we need to learn together. And the link for the registry is on the website, myotonic.org/registry. With this, thank you all so much. Thank you for being patient with me. Thank you for helping me to get through my moderation of this first webinar. And we look forward to seeing you all again in July when we hear from the next presentation. Thank you very much.